Have you ever wondered how scientists and researchers in the lab they isolate and visualize DNA? All the DNA is invisible to the naked eyes. Welcome back to my channel Genius Minds, and in today's lecture we are going to learn about a very important technique which is known as Agarose Gel Electrophoresis. So let's get started. Agarose Gel Electrophoresis is the very best technique used to visualize the DNA fragments. Basically, it is used for visualization and separation of DNA fragments. Now, in agarose gel electrophoresis, the word agarose is like a base and a very important material. It is extracted from the seaweed, and especially two important ones are Gracileria and Gelidium. So, these two seaweeds are involved in synthesizing the agarose. Now, let's understand the basic principle behind this technique. Now, as we all know, the charge on DNA is negative. So, the entire history works behind the charge particle. So, this negative charge is due to the presence of phosphate backbone. Now, the requirement for agarose gel electrophoresis are as follows. A basic requirement for agarose gel electrophoresis. We require agarose gel. We require electrophoretic tank, we require buffer, we require electric current. Now, as we all can see that in the name itself, the answer is hidden. Electrophoresis. Phoresis means movement. Electro means current. Under the influence of electric current, there is a movement of DNA particle. But how does it go? So, the movement is based totally upon the size of the DNA. So, the shorter fragments move faster and longer fragments take little time compared to shorter fragments. Now, what happens? I'll just show you the basic setup of electrophoretic tank. So, this is the basic setup of electrophoretic tank. We are having a power supply, we are having a gel, and to this gel we are having the combs attached to it. So, these are known as wells. These block-like structure are known as well. These are made with the help of a plastic comb, and this is the electric supply. The entire setup is placed in a buffer tank. So here we are going to make use of a buffer. Buffer helps in the migration of the DNA band, as well as it doesn't let the gel to get dried up, and also it maintains the pH. So buffer mostly we use is TAE, tris acetate EDTA, tris acetate EDTA. Also, it involves the fine moving of the DNA particles. Now, if you can see here, here is a negatively charged electrode which is known as cathode, and the positive one is known as anode. So, what exactly happens? As I told you, the charge present in DNA is negatively charged. So, according to the convention, it has tendency to move towards opposite pole, that is the positively charged anode. So, in the wells, the DNA sample is loaded. The DNA sample is loaded in the well with the help of a micropipette, and the electric current is switched on. Within half an hour or 45 minutes, it is seen that the migration of the DNA starts towards positively charged anode. And how are you going to visualize it? Because it is a transparent molecule, right? So you need a dye called as ethidium bromide. Ethidium bromide is an orange color dye which helps in the visualization of DNA fragment. The basic principle is that it intercalates between the DNA molecule. So whenever it intercalates between the DNA molecule, the dye is also moving along with the DNA particle. So once the gel is run, after that you are going to visualize it under an instrument known as trans-illuminator, UV trans-illuminator. So this instrument made use of UV light. UV stands for ultraviolet light. So whenever you are placing your gel under the UV trans-illuminator and switching on the UV, so UV has affinity towards ethidium bromide. The ethidium bromide glows orange in color. So wherever your DNA fragments are there, they will start appearing orange in color. Now also another point you can see, the shorter one can easily pass through the agarose gel. So that's why they reach faster when compared to the longer band. The shorter one reaches first when compared to the larger one. That's a basic principle. So this is all about the gel electrophoresis, and this is very important technique which is used in daily basis for extracting the DNA after the extraction of DNA in order to visualize this. And further experiments can be carried out by taking out your DNA band from this gel. That process is known as DNA elution. So that is another important process. So till here, it is all about gel electrophoresis. I hope you are liking my videos. If you are new to my channel, please do subscribe it, like it and share it as much as you can. Thank you very much.